If we look at epigenetics, there are many different levels. So in the next 3 lectures, we're going to show you each of these different components. This is coming from a review paper where they show the different level of epigenetics, starting from the very top, and that's the order we're going to discuss the epigenetics. The very first level is DNA methylation. This modification happens directly on the DNA. You can see in here. So yeah, so you can see here, the DNA is in a double helix. And some of these strands, if there is a C, at least in vertebrate species, most of the DNA methylation happens on the C nucleotide. So we know that human genome is A, C, G, T. Vast majority of DNA methylation in the vertebrate genome happen on the C nucleotide. And so this is a C nucleotide. It's normally there's also a sugar, on the, and the phosphate on the DNA nucleotide, but this is the C, and there's an enzyme which adds the methyl group. So previously this location, there's a carbon, and then the carbon is connected to a hydrogen bond. But if this hydrogen bond, so this is the carbon, if the hydrogen bond now is replaced with another methyl group, then this is a methylated C nucleotide. So 5, I guess on this this ring, there's a 1, 2, 3, 4, 5. This is on the 5. The 5 methylcytosine on this. Yeah, so this is a methyl group, then the DNA is methylated. For many, many years, people think that once DNA is methylated, it will be extremely stable. It will not get demethylated. For many years, it's believed that if you have, say, original DNA is like this, and if this group is now methylated, in order to create an unmethylated base, you have to make a DNA cut to cut this nucleotide out and insert another unmethylated C back into this place for it to be unmethylated. And so people think that once the region is methylated, it will not go through demethylation very often because, if you have to cut and change, it's a lot of work. But recently there are work that show, in fact, you don't have to really cut to replace. Instead, there are enzymatic reactions that can happen, which you can see through several biochemical reactions, eventually can make the cell back into a unmethylated state. And so there are different intermediaries there. But for many, many years, most of the research on DNA methylation just focus on this step. How can we map the methylated cytosine or the unmethylated cytosine? It's only in recent years that people are trying to map these, although these are very, very rare in the human genome. So most of the human genome, in fact, if a region has a C that's not methylated, so if a cytosine is not methylated, there are enzymes in the human genome that will try to add this C, and this C, once it's added, if it's not added, then this can also be very likely to be converted into a T if there's no C there. And so as a result, the human genome has a more frequency of having A and T's in the genome, and it's not 25% AT and 25% GC. It's higher percentage of AT and slightly lower percentage of CG, because if this region is unmethylated C, it's very likely to be, during replication, will be misrecognized as a T and be made into a T. So there are vast regions in the genome that are actually methylated, especially if they have C. So in the human genome, there are many, many repeats. Yes. Sorry, which one? Here or this one? Top or the bottom? The bottom? This tail is related to a nucleosome. This is not DNA. this is a pearl. This pearl is the nucleus of which we'll talk about at the end of this lecture and the beginning of the next lecture. This one happens on the cDNA, but this happens on the nucleosome. Okay, so most of our, if you look at the human genome, over 50% of our genome are repeats. And some of these repeats are coming from viruses. They come in and they can actually move around. They can, leave a copy and then they duplicate and then add another copy in different region of the genome. And that's how they become repeats. And DNA methylation is a very good way to basically bury it, to make it silence the repeat regions. Yeah, so most of the methylation in the human genome actually are found in the repetitive regions of the genome in order to silence them, to kind of, in some sense, if human find, well, in evolution, you can imagine if this virus is really active in the genome, it will do a lot of damage. But if you put the DNA methylation, it's like you try to bury it up, then it will not be moving around and creating damage to the genome. And so that's the majority of the DNA methylation. But there are several other regions that are potentially interesting. These are called the CpG island. So these are usually a few hundred base pair of DNA has very heavy enrichment of CNGs. Most of the region, if it doesn't matter, if it has a C, it will be methylated. But if it doesn't have a C, if it has a C but that's not methylated, it will be converted to a T already. Only in these regions, for some reason, the CGs are maintained and they can be either methylated or unmethylated. So so in these regions, so first of all, in human, so most of the repetitive regions are methylated, but otherwise it's these CpG islands. So these are CG-rich regions with high, CG density. Sometimes they are methylated, but sometimes they remain methylation-free. And so, in fact, majority of the human genes, if you look in front of this gene in the proximal promoters that are within the KB from the transcription start site, you will see that 70% of the human genes has a CpG island. In fact, if you find a CpG island without a gene nearby, there could be a new gene nearby that people didn't know, like a non-coding gene nearby. And so it's a very, very useful region. So if this region has a CpG island, it tells you there's something nearby that are potentially important. And by the way, when you look at the CpG, because human, very often the methylation happens, actually, they call it the CpG because there is a phosphate group between the nucleotide C followed by a G. In human, most of the CGs, methylations happen on the C that's followed by a G. For example, this C base, it's not followed by a G, so it's not often methylated. Okay, so methylation usually happens on the CG, and this reaction is symmetric. You can see because it's a CG on the top strand, it's also a CG on the reverse complement strand. And that C is also methylated in here. There are enzymes in the human genome that creates this methylation. But there are two types of enzyme. The first type, they do de novo DNA methylation. So for example, if you are in a cell state, previously it's not methylated, but then some regions become methylated. There are some enzymes that are in charge to do this. But also, there are enzymes that maintain the DNA methylation when the cell tries to replicate itself. And this creates the cell from one cell to the next cell, inheritance of DNA methylation. As we mentioned, if you have a heart muscle, when it divides, the next cell will also be heart muscle. How does the cell remember this? It's through partly because of this maintenance DNA methylation enzyme. So we can see how this is done. So for example, originally say we have the DNA that's double-stranded and methylation happened on the C base followed by a G. And on the other strand, the C is also methylated. Remember in DNA replication, each original parental strand can serve as a template to create the other strand of DNA, right? So this is the original DNA, and this is the original DNA. Yeah, so there it is. And they create the first strand, they each create the other strand of DNA. And then the DNMT1 enzyme will come here and say, if one strand is methylated, but the other strand is not methylated, it's gonna go ahead and methylate the other strand. This way, the two daughter chromosomes will have the same methylation pattern as the parent chromosome. Okay, and so that's how DNA methylation is inherited from one cell to the next cell. Questions on the DNA methylation so far?